Thank you very much, Ashty, for agreeing to give a Build a Cell seminar, and you can get started.
Fantastic. So, hi everyone. I'm Ashty Karim. I'm an assistant professor in chemical and biological engineering at Northwestern University. And I started my lab in January of 2025 of this year. But I've actually been at Northwestern since 2013. So I started here in 2013 in my doing my PhD with Mike Jewett. Um, and a lot of what fascinated me about synthetic biology was the idea that we could make any molecule we wanted using biological systems. And when I came to Mike's lab, he posed the idea, well, how about we do the metabolic engineering that you're familiar with and fascinated by, but outside of a cell? And that blew my mind that we could possibly do that. Now, I guess 12 years later, I'm still working in cell-free systems. And today I want to talk to you about data-driven design of cell-free metabolism and how we can study cell-free systems as a system and not just as a tool or kit to study biological mechanisms alone. So yeah, I'll get started. So when we think of cell-free systems and cell-free synthetic biology, there are many things we can use these systems to do to solve grand challenges in societal health and planet health. On the left, we have educational tools. We have the ability to use cell-free systems for decentralized biomanufacturing of medicines, therapeutics, and vaccines. We have the ability to make portable diagnostics for environmental contaminants or human health biomarkers. We have the promise of using cell-free systems for carbon negative manufacturing of chemicals, medicines, and materials. And what many of you are fascinated by, the ability to probe and design cellular function. And when we think of these cell-free systems and where they originated, we often cite, or before I get into that, one of the powerful things about cell-free systems is that we can power transcription and translation in vitro. So the idea is we take E. coli, grow it up, break it open, removing cellular debris and genomic DNA, and we're left with an extract. This extract can be supplemented with all of the components for transcription and translation and metabolism. So amino acids, NTPs, salts, cofactors, an energy molecule, a template DNA for proteins we want to make, T7 RNA polymerase. And through incubation, we can make proteins directly in the lysate. We can produce greater than a gram per liter in about 4 hours. We can use plasmid or linear DNA, these linear expression templates. In the bottom here, I talk about what I'm fascinated by is we can now treat this like a chemical reaction. So we have direct access to the reaction environment. These systems can tolerate growth toxic conditions, and they're compatible with high throughput assays. So we can make proteins and we can study proteins. And when we think about the origins of this, we often cite the Nuremberg and Matai experiment, where they discovered the first codon. So they created a cell-free lysate, they added mRNA, and then they made polypeptides. Oh no. But cell-free systems actually occurred much further back than this. So the first reported sort of cell-free system was reported in the late 1800s, early 1900s by Edward Buchner that showed that we could use press juice, as he stated, or extract to ferment sugar. So you could take cells, break them open. Here he used yeast and showed that by adding sugar without a living cell, you could convert that sugar into carbon dioxide and alcohol. He deemed that what was present were these zymases that allowed for this to occur, and it was the first cell-free system that received the Nobel Prize in 1907. And one of the reasons it received the Nobel Prize was actually because of a theory or sort of belief of vitalism. So this idea of vitalism was that certain chemicals, certain organic chemicals, could only be made by living organisms, and that there were inorganic chemicals that were primarily found in the earth as mineral deposits, but could be prepared by humans. And there was no way, it was because of life and this living force that you could actually make these types of molecules. And alcohols were one of them. And this is what Edward Buchner challenged, this idea of vitalism, that you could get fermentation to occur outside of a cell. So his discovery was celebrated as the resolution of one of the most famous scientific controversies. Um, was fermentation a vital physiological act of living yeast cells, or was it due to some purely chemical agent within the yeast cell? And he showed that kind of both sides were right. Fermentation is carried out by soluble enzymes in yeast extracts, but these enzymes were made by living cells. And so this spurred the study of what are these enzymes over the next couple of decades here, and a race to look and really birth modern enzymology of the time. This also sort of at this similar time led to the race to understand the central dogma of biology, from the nucleic acids being discovered to the structure of DNA being discovered. And that's where we get to the Mattei and Nuremberg discovery of the first codon in 1961. So here's the original paper looking at building a cell-free system and then dropping out or adding in components, and the radioactive C14 valine counts incorporated into the protein that you have here. And here's a graph over time of that protein synthesis. But actually, this cell-free system wasn't developed by them. The first cell-free system was developed 6 years prior by Joan Folks and Ernest Gill, where they created a cell-free protein expression system. The understanding of the central dogma was a theory at this time, and in their paper here, any proof of the part they play, so the central dogma plays, let alone its mechanism, awaits the preparation of an experimental system which can synthesize protein and on which an effect of nucleic acids can be demonstrated. Such a system has now been obtained. So what they did here was they built this cell-free system for translation using Staphylococcus aureus as their source strain. And a few years later, we get Lamberg and Zamenhof making the first E. coli-based cell-free protein synthesis system. Again, a complete system and then dropping out certain components to sort of test this extract ability to make proteins. And this served as the basis for the Nuremberg and Mattei experiments. About 10, 12 years later, we get Jeffrey Zubey creating the foundational protocol for today's widely used extract-based E. coli systems. So this is the protocol that calls for lysate clarification steps requiring centrifugation at 30,000 times G, and the first description of the S30 extract that we often refer to. And while we have many modifications today, most of that sources back to this protocol. In the early 2000s, we have Jim Swartz and colleagues looking at how we can actually make these systems more robust to make a lot of protein to study. And so looking at glycolysis and really the metabolism of the extract to start to optimize this for further performance. We have some really nice work from Vincent Noroa's group looking at actually going back to slow metabolizing sugar, so looking at maltodextrin as a way to fuel ATP synthesis for E. coli cell-free expression. And so I pose, and others have posed this, if we can control metabolism and we can control the central dogma, then we can recreate life. So this is a review looking at the pure system and really talking about cell-free synthetic biology as a way to understand, utilize, and expand functions of natural biological systems. Through purposeful design, transformation, and resynthesis of living systems, it can create and endow artificial life with unnatural functions. And this is cell-free protein synthesis is the core cell-free synthesis biology technique known as in vitro protein transcription and translation technology. Okay, so if we want to engineer metabolism, one way we do it, and what I've been doing for many years now, is looking at different ways that we can add in enzymes and study their function in the context of a lysate. So one way to do, or the ways that we do this are sort of twofold. One is through pre-enrichment of an enzyme that we might want to study, or post-enrichment. So pre-lysis enrichment is where maybe we take an E. coli, we transform it with DNA expressing a protein or enzyme of interest, and then we can grow those organisms up and make lysates out of them. Now we have a pre-lysate enrichment of these enzymes. Post-lysis enrichment is actually using the cell-free transcription translation machinery to synthesize proteins or enzymes directly in the lysate. So here we add either plasmids or linear expression templates and perform a cell-free gene expression reaction to express these. And then we can start to mix and match lysates enriched with enzymes to assemble enzymatic pathways. And instead of measuring protein concentration necessarily, we can start to analyze metabolites after incubation. So if we look at metabolism, so we have glycolysis here going into the TCA cycle. Transcription and translation reactions have been shown to utilize PEP, which I have highlighted in blue here, glucose, and many other starting molecules. And instead of transcription and translation, we can actually use these lysates to make molecules. So here in green, I have different molecules that can be made. And early on, from Mike Jewett's group, they showed that you could use make 2,3-butanediol from glucose, and you could achieve titers of greater than 80 grams per liter with peak productivities of 11 grams per liter per hour. And while cells have been shown to make more of that, that productivity is much higher than you could achieve in a cell. We've also looked, and a lot of my PhD work was looking at reverse beta oxidations, so reversing the fatty acid cycle to make alcohols and acids. And so in this mix and match approach, we can look at different enzymatic homologs for each step in this cycle, and we can mix at different concentrations and in different compositions of enzymes iteratively using machine learning to improve flux through this pathway and produce butanol. So that's what's being shown here. And what's interesting about this reverse beta oxidation pathway is it opens up it's like a module that can open up many molecules. So it makes many medium and long chain fatty acids and alcohols. So you can take an acetyl-CoA as an initiator, and you can stitch those acetyl-CoAs together in the cycle, and then you can terminate the cycle at several different points to get different types of acids and alcohols. And one of the things that we did was we looked at enzymes for each of the steps of this cyclic pathway in collaboration with Ramon Gonzalez's group at USF and Lanzatech, looking at natural proteins that do this and studying them in combinations. One of the things you can do in cell-free systems is start to play around with or understand synergies between enzymes in enzymatic pathways. So what I'm showing you here is that we took a starting pathway, and then for two enzymes, the first and second in the pathway, we screened homologs of each and looked at these pairwise combinations for combinations that perform better for the production of hexanoic acid here. We can then hop along this pathway and look at other synergies, 2 and 3, 3 and 4, and 1 and 4. And we can see which enzymes are most important for production of this molecule. And we can also look at metabolic flux through the cyclic pathway. So what I'm showing you is an experiment using SAMD-CoA MS. So the idea is you have a SAMD monolayer that you can pull down any of the CoA molecules in our lysates or in our cell-free reactions, and then you can observe what CoA molecules were pulled down. And over the course of a 20-hour reaction, using RBox, we can see different constituents of acetyl-CoA here, C4, C6, C8, C10 molecules. And you can see that over the course of the reaction, using different enzymatic homologs, we get different distributions of these CoA molecules. This allows us to start to observe different bottlenecks in our pathway, and then over the course of 20 hours, which CoA molecules are being sort of terminated in our reactions. We can also change the concentration of enzymes that we have to see that we start to build up flux to a point and create new bottlenecks. This gives us insight into optimizing metabolism and different biosynthetic pathways. And over the course of many different projects, we've actually been able to use cell-free systems, as listed here on the x-axis, using E. coli-based cell-free systems to test out pathway combinations for the production of different acids and alcohols. And then LanzaTech has actually taken these in their Clostridium autoathenogenum organism and expressed these biosynthetic pathways. And we see quite a good correlation, which was surprising to us. And really, if you dig into the details of the different works that we've done, it's cell-free systems serve as a good way to down select possible pathways and not necessarily select the best pathways, but remove pathways that should not be considered. Okay, so coming back to our map of metabolism, we looked at the fatty acid cycle and making these acids and alcohols, but we've also extended to more complex metabolic products. So looking at the production of isoprenoids, bisabolene, pinene, and limonene. And we did a similar thing where we were looking at enzyme homologs for each of the steps in our pathway here. And through iterative testing of different pathway designs, we can improve the production of these isoprenoids in the cell-free environment. We've also looked at taking amino acids and converting them into monomers for polymer production. So here we're taking phenylalanine and making styrene. And there are lots of things that we can do with testing enzyme concentrations and the physiochemical environment. So changing the pH here to increase the production of our molecule. But there's a lot of metabolism that hasn't been explored, and might be outdated now, but several people are trying to get to more complex metabolic products using cell-free systems. One, to understand how they work, and two, to actually potentially bypass some of the cellular limitations. One area of interest is actually trying to fix carbon dioxide using synthetic cell-free systems. This isn't a new idea. So there are several state-of-the-art formate assimilation pathways. So while there are 7 naturally existing carbon fixation pathways, many of these are inefficient. So there's been a lot of work to create synthetic pathways. Formate is interesting because it is a reduction product of CO2. And if we could take a formate and convert that into other metabolic products, we could potentially couple this with technologies that are really good at taking CO2 and making formate. And so in the last few years, we've looked at building this reform pathway. And the way that it kind of works is the idea is we could take CO2 and convert it to something soluble like formate and make acetyl-CoA, a key metabolic node. The idea is to create a pathway that's thermodynamically favorable, tolerant to aerobic environments, composed of a minimal number of enzymes, and devoid of that rate-limiting CO2 step, bypassing it in some way. So this reform pathway is what we came up with. And there are several different options. This is one. We were inspired by work really developing this OXC enzyme and engineering it, taking formal-CoA to glycolyl-CoA, and also inspired by the electrochemistry work looking at CO2 reduction to formate. The problem was all these core reactions are not known to occur in nature or haven't been reported to catalyze these reactions. So what we did is we went to engineer these. So all in cell-free systems, we can synthesize DNA variants, and we can produce proteins from them. So one of the enzymes, this phosphatidylase, naturally takes fructose 6-phosphate and converts it to acetylphosphate and erythrose 4-phosphate. We wanted to ask the question, are there phosphatidylases that allow us to take glycoaldehyde, or a chopped off version of this fructose 6-phosphate, and just convert it to acetylphosphate? So typically what we do is we look to nature first, identify enzymes and enzyme variants that might catalyze this reaction, and test them for initial activities. So we selected several different enzyme homologs from nature, and we tested them for this activity on glycoaldehyde. And we found one variant that actually did this really well. We then took this variant and looked at the substrate binding pocket, catalytic residues, and how substrates and products tunnel through the enzyme, and used this to sort of guide our selection of residues to mutate. We then mutated each residue here that I have. Each row is a different residue that we mutated individually. And on each column is 20 different amino acids. So every cell or every square here is one protein variant, a single mutation made. And then we tested activity on glycoaldehyde. And what we found was one mutation or one residue that seemed to be key in getting this functionality. We then went through iterations of site-saturated mutagenesis to improve this enzyme to a point that it was going to be useful in our pathway. We can then look at our reform pathway and engineer each of the enzymes in this step. So in the case of formate to formal-CoA, this ACS enzyme, we actually had to take an enzyme that takes acetate normally and get it to not take acetate. So really switch the substrate specificities. Engineering that enzyme as well, we can then take our original set of enzymes identified in nature that don't produce malate at the end of this or don't produce enough acetyl-CoA to make malate, and then our engineered version that actually can make malate. So it's non-zero now, and so now we can optimize and improve. So one of the cool things you can do in cell-free systems is you can start to iteratively optimize metabolic pathways and add metabolic modules that might be helpful. So what I'm showing you here is the enzymatic cascade, and we can use sort of design of experiments and simple linear regression machine learning to optimize our substrates, cofactors, enzymes to get better pathway performance. We can add ATP regeneration modules that have been published before, NADH regeneration, and metabolic proofreading enzymes to ensure that we get both carbon and energy flux through our pathway. Coupling that with electrochemical reduction of CO2, we were able to take formate and make malate at a relatively small amount. So this is not the end, but this showed that we could use both the protein engineering in cell-free systems and the metabolic pathway engineering in cell-free systems to take CO2 and convert that into a metabolic node. Okay, so if we want to optimize cell-free metabolism, or if we want to optimize transcription and translation, we often forget metabolism, and they're all connected. So our ability to produce proteins in these systems rely on all three of these processes. And so over the last several years, there's been a lot of really cool work combining modeling, artificial intelligence, and our ability to screen cell-free systems to optimize pathways, or really try to understand transcription and translation and the system as a whole. So I'm showing you three examples here that actually showed really good correlations between our ability to sort of define a set of reaction conditions and observe a protein translation product. What's interesting here is most of it is trying to basically predict and understand cell-free transcription translation and not necessarily optimize a cell-free reaction that's going to make a lot of protein for a little cost. And when we look at the cost of our cell-free transcription translation reactions, we have a standard Panox SP formulation. The energy cost of PEP is so high, and the cost to make our extract is also high, that for a milliliter of reaction, it costs about $4 to $5 per milliliter of reaction. And so we can go through hypothetical situations where we start to reduce those components in some way to get a reduction in price. But it's not as simple. If it was simple, we would have these really elegant works be able to find a solution. And so one of the things that we did was to go inspired by all of these ideas and really take our ability to generate cell-free data and our ability to use machine learning to engineer the function, and in this case, low cost and high yields of protein. And so a graduate student co-advised by Mike Jewett and I looked at many of the published cell-free reagent formulations, and in her hands, recreated each of these systems on the x-axis here, and then looked at the costs of reagents per gram of GFP produced on the left y-axis. And so those are the bar graphs. And then just gram per liter of protein. Those are the dots with error bars here. You can see that the Panox SP makes the most protein, but is in the sort of higher end of costs. And then we have these low cost formulations that make a little bit less protein. And so similar to the other studies that I showed you, we took consensus reagents that are used and did a combination of classical design of experiments and machine learning to test out 147 different reaction conditions or combinations to reduce the reagent formulations. And what's nice about this is we can actually reduce the cost per gram of GFP produced, but we start to hit bottlenecks or roadblocks that are hard to understand. And so one thing that we found is that by adding and removing reagents, rather than optimizing a fixed set of components, this was key to optimization of yield and cost. So we think we know what reagents are required to mimic cellular metabolism, but these aren't always the case. And as we start to optimize, we might need to add in new reagents, or if we start to remove reagents like nucleotides, so triphosphates, and add in monophosphate versions, this starts to get a little bit tricky. So this requires this iterative adding and removing as well as optimization. And what we found was a reagent formulation, which I'm showing you here in the first graph, compared to the Panox SP variant. And so we were able to find a formulation that costs $55 approximately per gram of protein produced that can produce over 2 grams per liter of protein. We then, in collaboration with Govind Rao's group at University of Maryland, showed that we could put this in a 1 mil bioreactor and increase the oxygen transfer to increase the productivity of the cell-free reaction itself. What I'm showing you on the bottom is actually taking our reagent system, sending it to 3 different sites, and each site, different operators could assemble these systems and produce 2 grams per liter of protein. And then on the right, I'm showing you GFP produced in different lysate backgrounds. So a reagent formulation is not one size fits all, and there's optimization that's going to have to be done as you change lysates. What's interesting, though, is then we took our lysate and we took the state-of-the-art, highest producing system and one of the lowest cost systems, and compared that to our RF-opt or our optimized reagent formulation to produce several therapeutic proteins of relevance. And in most of these cases, the optimized reagent formulation made the same or more protein than these other formulations. And the cases where that wasn't true, there's optimization that can and should be done. But this was very promising for us. And so the question now becomes, how can we create cell-free systems that last a week, a month, a year? How do we extend the longevity? Cell-free systems offer a high initial protein productivity, but die after about 12 hours. And if you were to re-add all of the reagents, we don't get double the amount of protein made. So why is that? The question becomes, can we decode metabolism and extend reaction longevity? So one of the things I'm doing in my lab, as I've started it, is try to think about metabolism, transcription, and translation as a system that needs to maintain some sort of homeostasis similar to cellular systems. So we often try to mimic cellular systems in our cell-free environment, but rather than mimicking that, why don't we create something specific to cell-free systems, unique to what our engineering objectives are to produce proteins for long amounts of time? The hope is this will open the door to many more applications than what we've already seen. And then we get to a point where how do we scale up and scale out these cell-free systems effectively? We need to further reduce costs of these systems, increase the range and capabilities of what these systems can do, and develop and demonstrate cost-effective applications. We can run cell-free systems at many scales, nanoliters all the way up to 1,000-liter scale. But the products, the things that we want to make using these at these scales differ because of many of the cost constraints that there are. So as I've started my lab, the goal of my lab is to build molecule systems and processes like metabolism using engineering, design, build, test, learn cycles. So the idea is we're going from molecules to systems to processes and working across all of these scales. To discover new enzymes, to develop biosystems and biohybrids. How do we take biological systems and merge them with electrochemistry, photochemistry, and others? And then how do we use the end goal in mind? So if we're going to create a bioprocess that operates at a small scale but regionally distributed, that actually changes our engineering objective at the molecular scale of what proteins we might want to make. So using this scalar approach, we're looking at trying to develop new technologies to biocapture our vast resource of carbon dioxide, bioconcentrate metals and minerals that are important for our modern technologies, and then bioconvert materials that we've created into others and more valuable products. So I want to end here to save lots of time for questions, but I want to, over this last several years, I've been able to have this beautiful collaboration with Mike Jewett's group, who's now at Stanford. And much of the work I told you was done in his lab when I was a research professor. And a lot of the work was done by these talented students and postdocs that I've had the privilege to work with. So the first RBox part was really led by Bastian Vogeli when he was a postdoc with us. Now at Exozymes, Grant Landwehr was a really talented graduate student who's now at a startup, Ridge Bio. And then Megan Olson is a current graduate student. A lot of the work was Department of Energy funded. Some of the cell-free metabolism for transcription and translation was DARPA sponsored. And then we have many collaborators, longstanding collaboration with LanzaTech and some national labs. And I'll end it there to take any questions.
Thank you very much. This was really awesome. There are some questions in chat. Can you see chat?
Let me see. I'm actually going to stop the share to be able to do that. So first one here, when you optimize pathway to increase production, what parameters do you change? Just DNA concentration or some other metabolic parameters of the lysate? So I assume this is early on when we're trying to make chemicals. So one of the ways that I described how we assemble the pathways is through this mix and match approach of enriched lysates, whether it's pre-enriched or post-enriched. And at that point, your design unit is no longer the DNA and how we construct the DNA or the concentration of DNA, but rather the enzyme or the lysate itself. So when we're trying to optimize or change parameters, we actually change the concentration of protein by changing the amount of that enriched lysate that we add to our reaction. In that way, we don't have to make new protein, but it's now a reagent that we have in stock. So the next question, could you please speculate what is the biggest bottleneck in optimizing capability? Is it energy, nutrient supply, ribosome stability, or something else? I wish I had a great answer for this question. And if I did, we would have these long-lasting cell-free systems that take over the world in the best possible way. I think the reality is it's all of the above. As soon as if you start to drop out or start to combat or mitigate any of the problems that you listed, you might see a minimal improvement, maybe statistically significant, uh, which suggests that that is a problem, but it's not the only problem. You start to create other bottlenecks. So it's this uh, vast complexity of the system itself, metabolism, transcription, and translation together, that become the problem that needs to be optimized. So what I would say is uh, we need to really tune lots of these levers simultaneously and then make sense of the data that we collect, whether that's and collect more data on protein synthesis rates, transcription rates, and metabolic rates. Oftentimes, we're just measuring an endpoint of any of those, and that's not giving us sufficient information to see what fails. Is it that ribosomes fall off? Is it ribosomes stalling, right? That's one potential part of the problem. Is it that energy dies or goes away, is used up? Is it that they've created byproducts that are now toxic? Do we now have actually metabolic insufficiencies or enzymes, lysate that becomes deactivated in some ways? I think it's all of the above that needs to be explored. Okay, does your optimization effort involve any attempts to compartmentalize subreactions in liposomes or in some other phase separation? Very cool question. Nothing that we did in our optimization used this sort of compartmentalization or organization strategy, but it's a very interesting space. You could imagine taking a lysate system or a cocktail of enzymes and put them in one phase and then another cocktail in another phase, right? Whether it's, if we're not looking at transcription and translation, that could be interesting as a way to separate different reactions to make them work more efficiently together. If we are looking at transcription and translation, that could be very interesting to mimic some of the things that we see in cells, but to create a cell-free organization system that is unique to cell-free systems. Very cool point. We did not look into that. How do you remove reagents from the mix, actually? Adding is easy, but selective removal is not, right? Yes, so one of the things I mentioned was adding and removing reagents. Really, this was talking about instead of adding 20 reagents, removing would be, let's add 16 of those reagents. So removing a reagent that we add, easy to do. Your point about removing something that's already in there or in a lysate, very challenging. So one way to go about that is to create a source strain that is engineered to remove some of these components, whether that's a knockout or tagged for removal later. Or one way we do this artificially is by adding inhibitors towards those. It's not perfect, and it is a challenging thing to do and may be necessary. Okay, so is bacteria lysate the way to go, specifically E. coli, or do you think we should look at more other extracts, eukaryotic or Vmax or other bacterial, or will it depend on the type of product and there's no one answer fits all? The last point is the answer there. I think it's beautiful that we can make all of these different organisms, but really asking yourself, what is the point or what is the purpose of using another organism? Is it if you're trying to make a lot of protein, sometimes these other systems are not optimized sufficiently to get a lot of a protein. E. coli has been the most optimized out of all these systems, and we often use that as the framework to optimize a new system. And what I showed here is this adding and removal and really going beyond the reagent formulations that have traditionally been optimized is key to optimizing E. coli further. This may need to be the case for these other bacterial lysates, and then or eukaryotic lysates. And then the use of them, I think, is we don't have a consensus on how useful they are. There's a hypothesis that has been shown in some cases and not in others that they can be useful to provide chaperones and other sort of effectors that might help folding function of a protein. So if you're making a eukaryotic protein, maybe you need a eukaryotic lysate. I think that has not been proven to the nth degree to a point that I agree necessarily, but it's worth exploring. Okay. You referred to 2015 article for the cost basis. Do these numbers still hold? Are there no updates given the cost of DNA has gone down reasonably and the break-even numbers you have different scales really been achieved or are these estimates? What's the largest cell-free synthesis scale achieved so far? Uh, yes, I cited a pretty old article to describe some of these costs, but in our latest work where we do the optimization, we to get the 55 uh, what is it, per $55 per gram of protein, uh, that we took up-to-date numbers to the best of our ability. Some of these numbers fluctuate, and DNA costs end up being very high. The table that I showed is a hypothesis, estimated amounts. And I don't think a good study has been done that I know of to look at those. As far as scales, most people aren't running above the 100-liter scale. There's been a lot to look at reproducibility across those scales. But again, you have to make so much lysate with some of our systems to actually look at those scales. I'm sure there are companies that are doing it, but I'm not aware of all of those. Okay. Is there a particular software package that you use to design new cell-free experiments based on the metabolic cycle that you are targeting, or are there a few standard protocols that you adjust manually as needed? So we typically have a standard protocol for E. coli systems. For the optimizations that you saw, we used definitive screening design, so design of experiments, adapted version of that, and linear regression models, many that are available through statistical packages like JMP. But in some of the other work that we've done with some of the machine learning, we're using a lot of the protein design tools that exist. But really with pathway design, you can't go wrong with standard design of experiments to start off. In the enzyme mutation work you showed, interesting that the top ones on the grid are histidine mutants. Something to do with the positive charges there. Have you looked into them in detail? Good eye. I went through that pretty quickly. Yeah, so what this opens us to, like if we're able to generate so many mutant variants, what can we pull out about the biochemistry or the enzymology that it is really cool? What we could do is start to look at some of these guided mutations based on what we find here, like you saw, or like you pointed out. We are also using machine learning to not necessarily look at the details, but find an engineering objective at the end of it, or match that objective. We have not gone in to explore some of this. We've looked at many different enzymes at this point, many that I didn't get to show you, but it's a really cool area that now you can do in the cell-free environment. Okay, the enzyme mutation work, no. To do a full study for cost characterization, currently all the costs are very high. Reagents per gram of product averages for thousands of dollars. Why? Are you aware of other efforts to bring down costs? So as you're doing the cost analysis, you can pull from look at all the reagents, see how much you can buy it for. So there are costs for small amounts, and then there are industrial reagent costs that are much cheaper, but you're ordering such large quantities that you can get the costs to be cheaper in those sense. There are some molecules that you cannot get any cheaper. One of the main reasons for us trying to find a way to use monophosphates instead of triphosphates for our nucleotides is to reduce that cost. That was one of the higher costs. Many of the early works looking at metabolism for powering transcription and translation looked at using these slow metabolizing sugars because they're a lot cheaper than phosphorylated energy sources. So The cost calculation really depends on where you're getting your initial numbers, which is why I think it's a hard thing to do. And then calculating the costs of lysate prep. I don't think anybody has one way of doing that. So it becomes challenging. But in each of the work where we talk about costs, we publish all the numbers that we're pulling from so that you could replicate whatever costs that we created or identify where there was a cost that we pulled a number that was more or less expensive than you could find. Are the major challenges for the cell-free still ATP, NADH recycling, phosphates? How do you think we can overcome other than the excellent works you presented here? Yeah, I think in metabolism, there are several things. The one most overlooked in my experience is pH. So these systems are very sensitive to pH. If you think about a cell, the cell membrane allows for regulation in a way that you can't get in your cell-free environment. So small changes in pH, compartmentalization can really help adjust things. And you have something very different in the cell-free environment. So systems tend to die as you decrease pH too much or you increase pH too much. And I don't think a lot of people look at the pH change of the reaction. Phosphates are probably linked to pH and other things that may be happening. And then ATP NADH recycling, a very important point. I think it still is a major challenge. And if you were to add new ATP regeneration or NADH regeneration, it doesn't fully recover the system. So the answer is yes, these are still major challenges. And I think the solution is really a more global optimization, changing many more things and measuring many more things. And then using some of the great modeling work that's been done in this space, combined with that large amount of data.
I think Tae-Sok has a
Yes, please.
in-person question.
Great talk. Lovely. I have some. Oh, by the way, I mean, I love the first part of your talk about the historic, aspect of cell-free. The reason I mentioned that one is, almost 30 years ago, I learned cell-free by the Dong Myung Kim and some other people,
Yeah.
working in the lab, when I was in Korea, and then they kind of teach some of that. And then the question I have is, you already kind of had a lot of answers to the question, talking about, economics and scale up, etc. And then I'm also an engineer. I'm already thinking about, new technology in terms of potential commercialization. And the first question is, you already keep the historic aspect of cell-free system and why it suddenly become popular. And that's my first question. This is my personal question. And second question is, you already answered some of the aspect regarding potential commercialization. We already have the great success that is a mRNA vaccine. That's a huge success in this technology. But there are multiple challenges already you touched. And then can you kind of give us some insight? What would be the limit in terms of their commercialization, in terms of product, and what else we can do in the future to make this technology more or broader, or more impactful, in the future?
Yeah, thank you for the question. Yeah, for me, understanding the history of the work that you do is an important exercise. And it's a field with a long history and a very important one. And if you look at that long history, why all of a sudden is it popular now? I think the same reason that a lot of our synthetic biology has become popular, the ability to read and write DNA, and then be able to start to use cell-free systems to study synthetic biology, understand synthetic biology and what we can do as a way of prototyping. So a lot of the work is done in prototyping and very little commercializing biomanufacturing in cell-free systems. Part of that, so getting to why that becomes like we're not seeing that as much, is these systems die. I think that is a key thing. They don't regenerate. Cellular systems do regenerate over time. You can run them longer and you can make more product over longer periods of time. Whether that's a protein or a chemical, it's all similar in that respect. There's a lot of work being done to identify which products can be made with the technology right now. So these are high value proteins, so therapeutics. And then there are several companies looking at enzymatic cascades that are more purified in nature and can last a little bit longer without being in the lysate system or the cell-free system. So I think one of the potential benefits that you don't see in other systems is this high initial productivity. So sustaining that initial productivity or being able to reactivate that initial productivity could make this a very beneficial technology for many products. And it really determines like what scale and where, right? If these can be lyophilized or dried, we start to open where it can be done very differently than our traditional systems that you might think. The other thing is when you think about proteins, you think about these large vats or reactors, right? Cell-free systems, I don't think will end up being done in those types of reactors. But like this, somebody from like those systems haven't really changed. Very little has changed. So you would recognize a 1950s bioreactor from a 2025 bioreactor. But what does that look like for cell-free systems? What if we could run small-scale reactions with high productivities and get the same protein products that you would get from these large vats? So I think there's a reconceptualization of what cell-free looks like in the field that will help with more commercialization. And as far as what products, I don't even know how to speculate on that. I think my area, what I find fascinating is pushing the system as a system and then trying to try everything so that somebody could then go take up and pull that up. But if we had a good question, maybe we'd have more commercial products or a good answer to that question.
Fantastic. You generate more product in the future from your lab. So congratulations again. Thank you.
I hope so. Thank you.
Sure.
Okay, one question. What tools, if any, do you use to simulate yields from cell-free systems, or does each one have to be prototyped and measured physically? A very good question. We don't do a lot to simulate our cell-free reactions. There are some basic ODE-based models that you can look at modeling transcription and translation, and there are a few metabolic models that exist, but they're somewhat low resolution, capturing all the reactions that are happening in cell-free systems rather than in cellular systems, which most of these are modeled after. So a lot has to be prototyped and measured physically. Luckily, we can run these in plates. We can run them at small scales. And it is a good technology for prototyping. But currently, we don't do a lot in simulating these yields, but other labs are.
And I think you got one more.
One more. To extend more on the enzymology studies, you think this is a better way to do studies of variants, full mutational scans in terms of costs? Have you done any comparisons? Very good question. I think it's an open question. Is it useful to do single amino acid mutations and do an entire saturated scan of a protein, or do something that's much less expensive? I think there's not enough. You could probably name on one hand the number of proteins that have been fully saturated individually to test to understand different changes in a protein. So I think there's benefit to do that. Is that the best way? Yeah, probably, maybe not. I think we need to know more before we can make a determination on whether you should do add some randomization there. I think what all of the work that I've done and worked with others on, what we've found is that the quicker you can get to combinatorial mutations, the better your result will be. So staying in the single mutant space for too long will be detrimental to you. So there are lots of ways to do there, but and I think AI is a powerful tool in this space. But what AI tools, when to use them and how to use them is the like current question.
And I think it's a question that we all need answer for, not just for biotech, but for using cell-free systems for practical applications, for synthetic cells, all the other stuff.
Definitely.
Yeah.
And there's going to be a combination of this more black boxy modeling and mechanistic understanding. And it together, I think, will solve lots of the problems in this area.
Yes. All right. If there are no more questions, I just want to say thank you so much, Ashty, both for an amazing talk and for answering so many questions. And thanks everyone for a great discussion.
Yeah, thank you. This was a blast. Hopefully I'll see many of you later. Yeah.
Yes. See you and have a great rest of your Monday.
Bye.
Bye.